So let's talk about Louis Pasteur. Okay. Louis Pasteur was a French physicist actually. He was a person of physics, chemistry also. Then he suddenly switched to microbiology because of various problems occurring at that time. The timing was his timing is 1822 to 1885, and he has done so many things in microbiology that he is remembered as the father of modern microbiology or father of microbiology itself. So he is called father of microbiology. We will discuss 4 or 5 contributions here. The first contribution is that he disproved the theory of spontaneous generation. Since the time of Aristotle, there was a belief that life begins from inanimate objects. I mean, non-living things. Life can start if you put some media, some kind of nutrition. Life automatically begins there. This concept developed because when in your life also you might have seen that some kind of food material, your bread, your meat, your milk is getting contaminated by different kinds of microorganisms. Some insects sometimes grow there, some fungus sometimes grow there, isn't it? So if you don't know, at this time you know that, okay, there are microorganisms, they can grow there. In the atmosphere there are microorganisms, but at that time, and even before the Leeuwenhoek's microscopic invention, I mean, when he first saw the microorganism, before that, people cannot think that, okay, life is there in the atmosphere also. That's why they thought that this is happening automatically, spontaneously. The life begins spontaneously. There is no need of anything. Life can begin from anywhere. Only you need what is that the nutrition. But the answer lies in this. If nutrition is required, I mean, does anything else require nutrition other than life? The answer was there. Only life requires nutrition, isn't it? We have to put some nutrient for plants or whatever animal you are growing, whatever thing you are germinating, whatever, isn't it? That requires nutrition. So though these people thought that life originates automatically from nothing. So this concept prevailed quite long until Louis Pasteur disproved it by his ingenious experiment of swan neck flask experiment. Yeah, this is also called theory of abiogenesis. But if you talk about the first origin of life, when life originated on the earth, it was a biogenic origin. Life originated from chemical substances by reactions, isn't it? At the very beginning. But after that, in the organic phase of evolution, life doesn't suddenly begin out of nothing. You require to put some propagules in the form of spores or seeds or zygote, whatever, isn't it? Or any living cell. Cell can be generated from a pre-existing cell, isn't it? That is the concept of cell theory also. So Lazzaro Spallanzani, Francisco Redi, these were the scientists who had almost proved that. Possibly you remember this experiment of Francisco Redi. He took 3 jars and within that he put some meat. This meat was preheated to remove all or make it sterile, to remove all organism. And then one of the jar was opened, uncovered, other was covered by some paper, and another was covered by muslin cloth. Muslin or muslin cloth. Okay. And then he incubated this for quite some time and he saw that maggots have grown in this a jar, in the open jar. Maggots have grown in the jar covered with muslin cloth because it is somewhat porous. So some amount of maggots, not many, not as much as the open one. And he did not find any maggot in the jar covered with paper. Though this is not foolproof experiment, maggots will grow here also because paper cannot, to that extent, yeah, I mean, it cannot stop the germs to enter at that level. However, when it is falling from the upside down, I mean, due to gravity, it will stop, definitely. So by this experiment, he said, Francisco Redi, he said that the life requires some propagules to begin, some egg, some spore to start with. The maggots, since they cannot enter into these jars, the eggs cannot enter into these jars, because of which maggots did not grow here. But here, because it was open, the eggs easily fell down on the meat. It got the nutrition and it germinated into a full-grown maggot. Okay, so he has proved this. But suddenly somebody contradicted, okay, you have stopped, possibly Francisco Redi also, that you have stopped the vital force to enter, not the eggs or the germs or these propagules. You have stopped the vital force to enter into those vessels. That's why organism did not grow there. Similar experiments by Lazzaro Spallanzani, who took media in a Erlenmeyer flask, meat broth. He took meat broth here, heated it to sterilize it, but this vessel was covered, sealed. This was sealed. No organism grew. The inference was again erroneous. I mean, it was the same thing. The vital force is not being entering. That's why it is not growing. Okay. So similar experiments done by Schwann, Snyder, and other people, they had almost proved that. The other experiment, possibly by Schwann and Snyder, what they did, they took the media, they allowed the vital force, so-called vital force, to enter to some extent to stop this controversy. They did their whatever possible way they could. That they took the media in a jar and they allowed air to pass in, but this air that was coming, it was heated. It was passed through heat. So it was a sterile air, you can say. And the media did not get contaminated. It remained as it is. So this was almost a good proof, I think, that you are allowing the air to enter. But again, other scientists said that no, you are heating and you are killing the vital force. Somebody passed this air through acid solution, and then also there was no contamination. Then also people said, no, you are killing those through acid solution. Somebody put a cotton over here, okay, cotton, Leadham possibly. He put the cotton in the pipe, that the cotton will stop it will allow the air, but it will stop the microorganism. And that is why that concept we still use, cotton plugging. The microbiological media, the vessel of the microbiological media is plugged by cotton. Still today, if you prepare media in conical flask, you will cover that flask with a cotton plug. Here you will put a cotton plug. That strategy is still used. Cotton plug allows the air to enter, but it does not allow the microorganisms to enter. Whether you put in a conical flask or in some culture tube, you have to put a cotton plug here. And there is a process of plugging this kind of microbiological apparatus. So after these many things, Louis Pasteur, ultimately he thought that I have to stop this controversy once and forever. And for that, what he did, He neither heated nor he put cotton nor he passed the air through acid solution. He simply allowed cold air to pass in. But the design of the vessel was such, swan neck, yeah. That design was specific and that actually helped him to prove this. So the design of the vessel was, the neck of the vessel was like this. Okay, the media was here, it was heated so that all organisms present prior to that will be killed. Okay, and he allowed to stand it for several days, no contamination occurred. तुम लोग ड्रॉ कर रहे हो कि नहीं, कुछ कुछ लिख रहे हो कि नहीं, जो जो चीज बोल रहे हैं. तो ये एक्सपेरिमेंट, दिस एक्सपेरिमेंट is the first contribution. What was the inference through this? He said that, okay, I'm allowing the vital force to enter. The force that you are saying, that vital force is essential, there is no such vital force. I'm allowing that vital force to enter. I'm not killing that through heat, neither killing through acid, neither I'm stopping it through by the cotton plug. Though there is no contamination. Means the microorganisms that are present in the air, they are somehow associated with dust particles in the air, or their particulate matter, they will settle down due to gravity. They enter this pipe, but they become trapped here. They become trapped here. They are not lifted up, and so they cannot contaminate it. That means what? The organisms are present in the atmosphere. That this thing was also clear. At that time, there was another scientist called Tyndall. You might be knowing Tyndall effect. He proved that there are dust particles, small microscopic particles in the air. When light comes, we can see those. Yeah, not travel along the light. I mean, light when strikes on those particles, light gets reflected, and you can see that. You can visualize that. That is what is called Tyndall effect, isn't it? So this was the first contribution of Louis Pasteur. The second contribution is germ theory of fermentation. You know, France is famous for wine and beer industry. And it is famous since long time, in 1850s, 1830s, 40s, since then. Fermentation process is an old process, that is true. We used to make curds and wines. Wine is an old, सूरा. यानी सूरा बनाने की प्रथा पहले से लोग जानते थे. तो कैसे हो, लेकिन how it happens, this was known that if you put a inoculum, I mean small amount of the prior culture or fermented product into a fresh medium, it converts into a fermented product again. Huh? But what is there inside, it was not known. In England, when people learned that, okay, curd contains lactobacillus, it is because of the lactobacillus and streptococcus that the milk is converted into curd, people stopped eating curd. So even if you talk these things at your home, your mom will not believe, if she is older, in my age period. तो उस समय कि नहीं ऐसा कुछ नहीं होता है, खराब नहीं हुआ, खा लो. मिल्क का टेस्ट बदल जाता है कभी कभी. आप अमूल का मिल्क खाओ, कभी नहीं बदलता. किसी कंपनी का मिल्क खाओ, वो लोग इतना स्टैंडर्डाइज करके रखता है इतना बढ़िया से कि वो कभी नहीं चेंज होता है. वो ऑर्गेनिज्म का उन लोगों का R&D टीम है ना, वो लोग देखता रहता है टेस्ट क्यों बदलता है. वहाँ जो ऑर्गेनिज्म ग्रो कर रहा है उसका नंबर में हेरफेर हो जाता है. लैक्टोबैसिलस की जगह कोई और स्यूडोमोनास, ई कोलाई या ईस्ट ज़्यादा हो गया तो वो कुछ और रिलीज करेगा वहाँ पर और उसका जो स्वाद है बदल जाएगा. हलुद हलुद टाइपेर हो तो दूध हो जाए ना कि कार्डो हो जाए, दूधर ऊपर डिपेंड करे अनेकटा. तो मोषेर दूधेर एकटू हलुद हलुद दिए थाके, बेशी छाँच थाके बोले. तो এটা কম্পোজিশন অফ দি মিল্ক ইজ ইম্পর্টেন্ট. that it allows some other organisms to grow there. So this was the period when these things were revealed. First of all, the fermentation occurs because of some microorganisms. Because Louis Pasteur and others also, they were seeing that in the fermenter, in the media, the vat where fermentation is taking place, that vessel, when you study that sample, you are getting one type of microorganism more than the others. Why this is so? Why they are more in number? They are growing there, that's true. Others are also growing there, and possibly they are the main culprits. I will not say culprits, they are the main individuals who are converting that, who are doing that process, job. This conclusion will come in your mind. So the change that is taking place during fermentation, the substrate being converted into a product, that is being done by this individual which is more in number. Every time when you study, you find the same individual more in number. So now you will definitely be confirmed that, okay, this one is the main organism which is causing this change to happen. So organisms cause fermentation. This is now your conclusion. That is what that was known. And when your fermentation gets spoiled, when it is getting spoiled because of, sometimes you try to set curd at your home, every time you will not be able to do that, है ना? दूध दही जमाने का कोशिश करो, हर बार दही वैसा नहीं बनेगा जैसा तुम, क्योंकि तुम्हारे पास जो ये जोरन जिसको बोलते हैं इनोकुलम, वो बार बार चेंज कर रहे हो, एक से लेकर दूसरे में दे रहे हो, दूसरे से फिर कहीं और से मिल गया उसको दे दिया, तो पहली बार तो उसके जैसा होगा, दूसरा बार भी उसके जैसा होगा, तीसरा बार जब तुम देर कर दे रहे हो ना देने लेने में कि किस टाइम में तुमको निकालना है और फ्रेश मिल्क में देना है, ये एकदम स्टैंडर्डाइज होना चाहिए. तो इसको बोलते हैं स्पॉइलेज, फूड स्पॉइलेज, मटेरियल स्पॉइलेज होता है ना, लकड़ी में फंगस ग्रो कर जाता है, स्पॉइलेज. प्लास्टिक तो बहुत रेजिस्टेंट, बेल्ट वगैरह जो होता है लेदर का प्रोडक्ट, उसमें भी फंगस टंगस ग्रो कर जाता है, सड़ने लगता है, isn't it? So spoilage, मटेरियल का स्पॉइलेज फैक्टर है. Spoilage due to microorganisms, not by atmospheric spoilage. I'm talking about. কিছু প্রবলেম নিচেই মনে হচ্ছে, যাই হোক চলে আসবে. So spoilage was taking place. Spoilage is a big factor in food microbiology. Those people study spoilage. If you keep meat at your home, not in the refrigerator for some time outside, it will get spoiled. Putrefaction will take place. Protein degrading bacteria and fungi will grow there. Same is the case with the fish and any other food material. In summer season, if you keep your dal, pulses, or vegetables what you have cooked, yeah, just in 3-4 hours will be enough to make it spoiled. There will be a sour taste in that, and that is because of some kind of streptococcus or kind of bacteria. Bacterial spoilage, fungal spoilage. This is a big factor. So that's why we use preservatives in different food items so that it can be kept for longer time, shelf life. To increase the shelf life, you have to put in the refrigerator or in the freezer, and at the same time you can put some microbial, antimicrobial but edible substances, less toxic or neutral substances which can inhibit microbial growth, okay, but will not be harmful. That kind of preservative is also required in research also. You need to find out new preservatives. And preservative have their own taste. That is unwanted. तुमने एक चीज को खाना को प्रिजर्व किया, उसमें प्रिजर्वेटिव दे दिया, वो प्रिजर्वेटिव उसका स्वाद बदल दे रहा है, तो वो भी तो अनवांटेड है. हमको तो वही स्वाद चाहिए. समझ रहे हो ना? ये सब फैक्टर है, बायोलॉजिकल फैक्टर है, इसको हल्के में लेना ठीक. तोरा की कोई चीज रे माइक्रो मोबाइल लिए ओवर स्मार्ट কি করছিস, যেটা বলছিস সেটা ঘাঁটছিস নাকি অন্য কিছু? ওট ওখান থেকে, ওট এখানে বস, একজন ওট ওখান থেকে এখানে বস. ঠিক আছে, তো germ theory of fermentation says what? That fermentation occurs because of some germs, means some microorganisms. This is this second contribution. Third contribution is this germ theory of diseases. Germ theory of diseases. Before going into that, one more thing that is associated with this ferment theory of diseases. Before going into that, one more thing that is associated with this fermentation is the process of pasteurization. Pasteurization. You have not heard about that? Pasteurization? Why? हाँ? क्यों नहीं सुना? तुम्हारा दूध जो खरीदते हो उसमें लिखा रहता है पैकेट पे पास्चुराइज्ड मिल्क. Pasteurization, what is that? Developed by Louis Pasteur. कि ये जो spoilage microorganisms की बात कर रहे थे, spoilage microorganisms, to remove those microorganisms from either wine or the milk. Milk, in the dairy industry, milk is converted into many different products, like the curd, yogurt, cheese, etc. So other organisms start growing there. So he developed a process by which the property of the milk will not be compromised. I mean, its constituents, not only dense, chemical constituents, amount of protein, vitamin, or minerals, or lipids, whatever there in the milk. Milk is a complete nutrition, it is being said. So we heat it for several times until it boils and it becomes, viscous. We continue boiling it, but we destroy many of its ingredients. And also the healthy microorganisms. Microorganisms all are not bad. Those microorganisms should be present for that dairy, those products, to make those products of that quality. If you are removing one or the other, the quality of the product will change. This is very minute change you cannot easily observe. There are people who work in different fermentation industry just to taste it, just to taste the wine, the little bit difference of wine they can identify. है ना, होता है ना? So that's why the pasteurization is a process. Let me tell you, pasteurization is a good process. You can write a note on pasteurization. Might be this kind of questions are asked. Short question or maybe 2-3 marks question. Pasteurization, at that time what he developed, he said that to heat milk or similar products to 62.8 degrees centigrade for 30 minutes. 62.8 degrees centigrade, 145 Fahrenheit, or 62.8 degrees centigrade for 30 minutes will remove the harmful microorganisms. The harmful microorganisms, spoilage microorganisms, as well as there are some which are pathological, like the Mycobacterium tuberculosis. This was also there. 2 microorganisms specifically he wanted to remove, or other people after him also, they wanted to remove Mycobacterium tuberculosis. The bacteria responsible for tuberculosis, TB, the tubercle microbacteria. And the other one is Coxiella burnetii, responsible for Q fever. These 2 bacteria get removed at 145 degrees Fahrenheit if you treat it for 30 minutes. It is too less. For 62.8 degrees centigrade is not too much. In 30 minutes it kills Mycobacterium tuberculosis as well as the Coxiella, but it doesn't kill the Lactobacillus, the Streptococcus, there are others also, Bifidobacterium, E. coli, Pseudomonas, Alkaligenes. There are many other bacteria in the milk. They have to be kept. They are the natural inhabitants of the milk. They will be changing the character of the milk when that fermentation process is going on. The amount of aldehyde, the amount of acid they are forming, that affects the taste and quality and the fragrance and the texture of that product. Okay? So pasteurization is one thing which is associated with the germ theory of fermentation. In that heading, you can write that. Then germ theory of disease. Pasteur had earned fame in France, that the government of France thought to give him a project that you find out why our silk industry is getting affected due to a disease called pebrine disease. What is causing this pebrine disease? Pebrine disease. Pebrine disease. What is the exact reason of pebrine disease? It was not known at that time. So Pasteur was given this project or task to find the causal organism. Because of this, your silk becomes, the silkworms, the larvae of the silkworms, they die. And your silk becomes, the silk industry gets affected, the yield decreases, they cannot form the cocoon, isn't it? So Louis Pasteur found it that, okay, the pebrine disease is taking place because of the protozoa, not a fungal disease, not a bacterial. It is because of the protozoa. It's causing this pebrine disease to occur, killing the larvae of the silkworm. You got it? Chart number, development of attenuated vaccines. Development of vaccines, attenuated vaccines. Louis Pasteur developed the process of making vaccines. In chicken cholera, he first made the vaccine of chicken cholera. Okay, what he saw that when a chicken does it look like a chicken or what? When a chicken is injected with old culture of chicken cholera bacteria, it becomes resistant against cholera. When a chicken is injected with an old culture, 8-week old culture he used, when it is injected with an old culture of the bacteria, and then when again the same chicken is injected with a fresh culture, it does not die. But another chicken who was not injected with the old culture prior will die on the inoculation or injection of the fresh culture. What does this reveal? What is the inference that could be drawn from here? That the old culture is giving protection against the particular organism. So by using old culture, we can immunize an organism, even human being, just like pox. Yeah. In the pox, the story was a cross protection. I mean, variola virus was being used for protecting vaccinia virus, isn't it? Little bit different. That was cross protection. Cowpox is protecting smallpox. Little bit different. So that was also interesting, that one step advance, in fact, that if cowpox can protect smallpox, then smallpox will better protect smallpox. Vishe vishe vishokho hoy, vishokhoy hoy, ये कथाटा আছে, विष ही विष को काटता है, ऐसा एक कहावत है ना? हाँ, क्या बोलते हैं? हाँ? मुहावरा है, विष ही विष को काटता है. कोई संस्कृत में होगा ऐसा श्लोक, लोग हमको नहीं मालूम. अब बंगला में बोलते हैं विषे विषे विषोक्षय है. ये सांप के काटने पर बोलते हैं सांप का ही ये दिया जाता है, वो सांप का वेनम तो नहीं दिया जाता है, उससे प्रोड्यूस किया हुआ एंटीबॉडी दिया जाता है, तो इसलिए लोग बोलते हैं. तो यहाँ भी क्या हो रहा है, हम एंटीबॉडी को ही, हम जो उसका एंटीजन पहले डाल दे रहे हैं ताकि हमारे बॉडी में उसके अगेंस्ट एंटीबॉडीज बन के पहले से तैयार रहे. वैक्सीनेशन का कॉन्सेप्ट तुम लोग का नेक्स्ट क्लास कब है, कितना बजे? अभी है 12টা থেকে আছে. এমডি, আচ্ছা, এটা তো মুশকিল হয়ে যাচ্ছে. একটা করে 1 ঘন্টা কমই পড়ে যাচ্ছে। তো যাই হোক, আমি তাড়াতাড়ি তো কি বললাম যে ওল্ড কালচার of an organism, bacteria here, chicken cholera, it gives protection against the same organism. Because in the old culture, because of the passage, culture means what? You are putting the organism in a medium. It is not its natural environment. The natural environment of the chicken cholera is the chicken's body. Okay, so if you are putting an artificial environment, say the artificial environment is a room which has everything, every facility. I mean, you get automatically when you ask, give me rasgulla, chalayega, give me pantua, chalayega, give me singara, chalayega, ठीक है, show me some film, कुछ भी मांगो चलाएगा। तो तुम्हारे अंदर का जो क्वालिटीज है, बाहर लड़ने की क्षमता, वो खत्म होने लगेगा। कैसे कमाते हैं, कैसे लोग सामान बेचते हैं, या फिर नौकरी के लिए क्या क्या करना पड़ता है, सारा कैरेक्टर धीरे धीरे खत्म होने लगेगा। अगर आपको उसमें आपको पूरा मिनरलाइज वाटर, स्टेराइल वाटर, बढ़िया बढ़िया चीज़ एकदम प्योर दिया जाए, नो इन्फेक्शन, सो योर बॉडी विल गेट ग्रेजुअली, आई मीन, इम्यूनो कॉम्प्रोमाइज्ड। ऐसा ही होता है शहर के लोगों का, जो ज़्यादा मिनरल वाटर पर डिपेंड करते हैं, उनका जल्दी से इन्फेक्शन हो जाता है। और जो गरीब आदमी जो कहीं का भी पानी पी लेता है, मिट्टी के साथ रहता है जुड़ा हुआ, उसको इम्यूनाइज, पहले से इम्यूनाइज्ड है। दैट मीन्स योर इम्यून सिस्टम, दे आर सेल्स, दे प्रोड्यूस एंटीबॉडीज, सम इज सेल डिपेंडेंट, सेल मेडिएटेड, दे नीड टू बी ट्रेंड। एक ट्रेनिंग का जरूरत है। हम ये सब बात तुम्ही लोग से बोल रहे थे ना। तो इसीलिए ओल्ड कल्चर में क्या हो रहा है ना, उस अपना जो प्रॉपर्टी है चिकन कोलरा का, वो धीरे धीरे खत्म हो जा रहा है, वो एटेन्यूएटेड, मतलब पतला हो जा रहा है। एटेन्यूएशन मीन्स डाइल्यूट हो जाना, धीरे धीरे रिड्यूस हो जाना। तो द एफिशिएंसी टू कॉज इन्फेक्शन हैज बीन रिड्यूस्ड, एंड बट द एंटीजन इज देयर। द एंटीजन विच इज ए प्रोटीन और ए ग्लाइकोप्रोटीन, व्हाटएवर, दैट इज देयर ऑन द सेल, एंड आवर बॉडी रिक्वायर्स दैट। हमारे शरीर में जब वो घुस रहा है तो वो एंटीबॉडी बना रहा है, जस्ट लाइक द कोवैक्सीन वैक्सीन अगेंस्ट जो SARS-CoV-2 में। कोविशील्ड वाज ए जेनेटिकली मॉडिफाइड वैक्सीन, कोवैक्सीन इज एटेन्यूएटेड वैक्सीन। उसको भी हीट किल या फिर कोई केमिकल से भी किल कर सकते हैं, देखना पड़ेगा, फॉर्मेल्डिहाइड वगैरह से भी करके बॉडी में दिया गया है। उसका जो स्पाइक प्रोटीन है वो हमारे बॉडी में गया है, होल कोरोना नहीं। ठीक है, तो ये था वैक्सीनेशन।